So when we talk about viruses, this is the very last topic of your chapter 1. In 1852, a Russian scientist discovered that certain diseases could be transmitted by agents that, unlike bacteria, could pass through very thin, fine filters. This was the first evidence for the existence of viruses, that there are agents present in this world that can even pass through the very small and fine crystals, because they are so tiny particles, even much smaller, 1,000 times smaller than bacteria, and are on the boundary between that what we think of as non-living and living. They're on the boundary. If we have living organisms on one side, non-living on the other one, they are present in the middle, the boundary. Unlike prokaryotes and eukaryotes, viruses do not have a cell structure. They don't even have organelles, because if they did have any organelles, such as chloroplast, mitochondria, ribosomes, or anything else, they would have carried out many of the characteristics that are in a living organism. Viruses are a very small 20 to 300 nanometer infectious particles which can replicate only inside living cells. It consists of a molecule of DNA or RNA, which is surrounded by a protein coat, and outer lipid envelope may also be present. They are not surrounded by a partially permeable membrane containing cytoplasm with ribosomes, and they are very simple in structure. Now, there are two types of viruses. If a virus contains a DNA, now virus can contain DNA as well, RNA as well. But if we talk about eukaryotes and prokaryotes, they all have what? DNA or RNA?
DNA.
Exactly. If a virus contains DNA, we call it DNA virus. But if a virus contains RNA, it is known as a retrovirus. Retrovirus, which is a very harmful one. An example is the HIV that causes AIDS, the human immunodeficiency virus. It is a self-replicating molecule of DNA or RNA, and there's a protective coat of protein molecules as well that surrounds the whole virus molecule, which is known as capsid. And that capsid is made up of capsomeres. It's something like this. It's a protein coat that makes up the surrounding of a virus. And inside the virus, we then have this molecule of RNA. So RNA is present inside this capsid. Capsid is this whole layer, the outermost one. And this capsid is made up of these small ball-like structures which are known as capsomeres. Got it?
Yeah.
Some viruses, a membrane-like outer layer called the envelope, is made up of phospholipids. Figure 1.36 shows the picture of a virus with an envelope, and viruses typically have a very symmetrical shape. What does symmetrical mean? Can you explain me the whole structure over here that you can see?
So there's a protein coat, and then there's like spikes of protein on the coat, and then there's a capsid,
Mhm.
and then inside there's the DNA or RNA.
Good. So we have an envelope protein, an envelope, a capsid made up of capsomeres, and then the substance that we have the RNA, then comes the capsid, then comes the envelope, and then come the protein that are present on the envelopes. Viruses range in size from about 20 nanometer to 300 nanometer, about 50 times smaller than average than bacteria. All viruses are parasitic. What does parasitic mean?
They need like a host to survive.
No, host is a human body, the one which gets infected. What does parasitic mean? That viruses are parasitic, parasites.
Like they get their nutrients from the host and not from like
Good,
themselves. Like they
good.
make their own.
They are infective, and they can cause infection. The virus DNA or RNA takes over the protein synthesizing machinery of the host cell. This is the very first job that a virus does when it enters the host cell, is take over the protein machinery, which then helps the virus to make new kinds of itself and new virus particles as well. And that is the end of your chapter of viruses. There's one small thing, a very small video that we have to watch regarding viruses, and then we are good to go. We'll start uh, you know, some topical past papers, some end of structured questions as well. Let me show you.
Viruses are non-cellular infectious agents that can infect and replicate replicate within cells of organisms such as plants. Viruses are non-cellular infectious agents that can infect and replicate within cells of organisms such as plants, animals, and bacteria. Today, we're gonna take a look at the structure and the features of a virus. Subscribe to the channel if you don't want to miss out on new science videos. Even though you may often read in older school books that a virus is not classified as an organism, it is still an ongoing debate whether they can be considered living or non-living. There is in fact an increasing number of scientists saying that a virus has indeed characteristics of a living thing. However, per definition, a virus and its processes such as replication or protein biosynthesis rely on the metabolism of the host cell. Viruses come in many shapes. There are helical, polyhedral, spherical and complex viruses, such as the bacteriophage. There is no such thing as a general structure, but there are distinct features that are shared between some groups of viruses. Some components are found in every virus, some others are exclusive to certain subtypes. The most important component of a virus is its genome. Any virus possesses a nucleic acid that carries the genetic information required for the synthesis of essential proteins. The main function of a virus is to replicate the genome to produce more of its kind. In parts, the replication machinery of the host cell is exploited to this cause. Viruses are often classified according to the type of nucleic acid. Some viruses have a genome out of single-stranded DNA, others possess double-stranded DNA. There are also viruses with single-stranded RNA or double-stranded RNA as genetic information. The genetic information is surrounded and protected by the capsid, sometimes also referred to as protein code. Any virus has this feature. The protein code is made up of small repeating subunits called capsomeres. These protein building blocks are encoded within the viral genome. In addition to the capsid, not all, but some viruses have an external lipid membrane known as the envelope. The envelope lipids are not encoded by the viral genome. Instead, the lipid envelope is derived from the host membrane on the way out of the cell. Further, within the envelope of some viruses, there may be glycoproteins. The genetic information encoding those lies within the viral genome. The glycoproteins may facilitate binding of the virus to receptor sites on the host cell or assist in entry into target cells. A few viruses also have their own enzymes. The retrovirus, for instance, contains a reverse transcriptase. Just like the envelope, enzymes are also not a universal feature of all viruses. Overall, the structure of a virus is simple but effective. The genetic material and protein code work together to allow the virus to enter host cells, replicate inside, and spread to new cells, ultimately causing disease. More interesting and topic-related videos can be found here. Make sure to subscribe to the channel.
All right. So let's read the whole summary of your chapter number 1. How did you find it? Was it overall tough, new, difficult, easy? How did you find it?
I think some parts were okay, but like some parts there are just a lot to remember.
Exactly. No, it is. Yesterday's class was too much. I know that. Okay. I should have divided that into 2 lectures, but I think you still understood and you have drawn the structures. I saw your work as well. You don't need to worry. You've got it all. So the basic unit of life is the cell. And the simplest cell are the prokaryotic cells, which are thought to have evolved before and given rise to the much more complex and much larger eukaryotic cells. Cells can be clearly seen with the aid of microscopes. Then we talked about 2 types of microscopes. One of them is a light microscope. One of them is an electron microscope. And electron microscope also has 2 types, scanning microscope and transmission microscopes. Then we have a formula for calculating the actual size as well. Then we talked about the partially permeable cell surface membrane, the DNA, the function of DNA, the nucleus. Then we also had some organelles, including endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, mitochondria, centrosomes, centrioles, cilia, and cell walls made up of cellulose, chloroplast, vacuole, Prokaryotic, we talked about, we have done prokaryotic cells today and viruses today, right? So prokaryotic cells lack a true nucleus. They don't have a nucleus. They have smaller 70s ribosomes than eukaryotic cell. And they also lack these membrane-bound organelles. Their DNA is circular and lies free in the cytoplasm. Viruses do not have a cellular structure. They are extremely small and simple. They consist of a molecule of DNA or RNA and a protein coat sometimes as well as an outer envelope. Got it?
Yeah.
Do you have this book?
I have the online PDF, but I don't have the book.
Yeah, same here. I have the online one as well. Okay. Solve these 3 MCQs. I'm sharing them in the group. And then that is all for today. No other written work for today. Clear?
Okay.
All right. Goodbye.